keep in mind that DNA replication is DNA replication and protein synthesis is protein synthesis. Tend to confuse them or mix them together. It's two separate parts of these notes, okay? So protein synthesis occurs in two steps. It's transcription and translation. And we're going to cover transcription today and translation tomorrow or Wednesday. So transcription. Has anybody ever heard of a transcriber? What are you doing if you're transcribing?
Like writing, rewriting.
You're rewriting something. A court reporter does that. They take messages during like a court in like a code. And later they take all those codes and they transcribe the message into like a written word. Okay? So the same thing happens. There's a message where?
In the protein?
Nope.
In the DNA
model. I mean the DNA strand. And it's decoded in order to create proteins. That's how it starts. Now, if you look at this picture, if I gave you this test, this picture, which can happen, right? And it's a test, where are we right here? What is this whole circular thing? A cell. And this blue area is? Nucleus, okay? And we know this is DNA because it's labeled, but let's say I didn't put a label. How would you know that that's DNA?
Because it's double helix.
Because it's a double helix. It's double-stranded, okay? So what's the first thing that happens here?
DNA unzips.
DNA unzips, okay?
RNA?
How? No. Because the DNA is like a template for those nucleotides to come in and form a strand. So if DNA's strand has these letters, A, T, C, G, A, what's RNA going to be like? Raise your hand so I can hear from somebody. Yes. UAGCU. Now, I saw a lot of you getting confused. So probably the best thing you could do to avoid making a mistake is do this. TAGCT, yes? And then come back and everywhere you see a T, turn it into a U. Because a lot of times people will take where the T is and put a U. Okay, we know the corresponding DNA or nucleotides in DNA. If you're confused to follow the pattern, make it DNA again. TAGCT, and then go back and erase all your T's and turn them into U's, because RNA does not have T. Good. Yes.
Wait, so the order...
Another example, if I gave you this strand was DNA, then RNA would be the corresponding basis. So depending on the strand I give you. Now, technically I could give you an RNA strand and tell you what strand from DNA was it made from. So I could do it backwards, right? Okay. Now, once RNA is made, it's ready to go make protein. So what's it going to look for? What do you need in order to make protein? We got DNA. It made RNA. What's missing? No. What makes protein? Ribosomes. So it's going to leave the nucleus and search for a ribosome, which is right here, and start the process of making of protein.